Okay, so we're going to launch into cell bio. This is where we belong. But first, what am I supposed to do? Jokes. Look, I already got you guys trained like Pavlov's dog. What did the mom chameleon say to her nervous kid on the first day of school? Don't worry, you'll blend right in. See, I told you after a while you get in the habit of it. It uses another side of the brain, that's why. We'll get there, don't worry. It's actually true. All right, what kind of bird can lift heavy objects? A strong bird, that's true.
Big bird?
Not big bird, that's what I was going to say. A big, strong bird, right? A crane. That one actually was written by my 12-year-old, so it's pretty good though. I'm so proud of that. She's also good at math too. All right. So this is the last lecture that's review, and then we're going to launch into new material. Some of the new material we'll get to today. We'll finish it up on Wednesday. We're gonna get as far as we get. So don't stress. If we don't make it through all the material, then not all the material will be on this first exam. And that's where we'll pick it up on exam 2. Okay, and I'll make that very clear so you don't have to, stress. It's not going to be like, catch all that. We'll just go the pace that we go and we'll get caught up. We got all semester long to get caught up. And then wipe off the So cell bio, many of you have had this lecture or these lectures before, but this is sort of a review to get us all on the same page. So cells, what are they? Well, they're the building blocks of life. And this, backing up one slide, this picture on the upper left, what is that? That's an artist's representation of what? I don't know, something from like, Tron or, that is the inside of a cell. Okay. So you've got the nucleus, right? You have intracellular organelles. It's a fascinating structure, and it is this basic building block of life. That's where we have defined, next Monday, you will know correctly, that life begins at the level of the cell. Right? And that's how you'd answer that question on an exam. Of course, there's all sorts of little caveats that we talked about, and a couple of you in this class actually gave really, really good answers on that. But for our purposes, we say this is the building block of life, and we can't have living tissue if it's not cellularized. Now, down here, does everybody know what these cells are down here?
Those skeletal cells?
No, that's a good guess though, because you see striations. These are actually called Hela cells. Hela cells are named after a woman named Henrietta Lacks, and they're a spontaneous mutation of a cell line that was a cancerous cell. These were cervical cancer cells, and the physician back in the day, this is in the early 50s, 1950s, decided they were so fascinating to watch them divide uncontrollably that he started doing experiments on them without her permission. And she passed away. And then he started handing them out to his colleagues. And they're one of the most popular cancer cell lines used even today, Hela cell lines. And this is in California. And so the family sued. And all that paperwork you have to fill out now when you go see the doctor? This is part of the reason. Because the Supreme Court of California decided that once the cells leave your body, they no longer belong to you. So she didn't have rights to those cells. Okay? Whether you agree or disagree with that, this isn't a philosophy class. I'm just trying to give you a little background of what has happened within medicine. And we've been playing around with cells for a while, and even cancer cells since the 1950s. And things have changed a little bit. The rules have changed a little bit. Well, the rules changed because of things like this, but Hela cell lines are named after a patient, Henrietta Lacks. Okay? They're really interesting cell lines, and we're going to see how other cells in our body, not necessarily cancer cells in this class, but we're going to look at muscle cells, we're going to look at fibroblasts, we're going to look at skin cells, we're going to look at smooth muscle cells, We're going to eventually get to cardiac cells. We may not get there today's lecture on Wednesday. And you'll cover cardiovascular much more in depth in 202. Okay? So why do we start with cells? Well, they're the building blocks of our body. That's what we're made of. And they're specialized into different regions. We didn't really start appreciating the cell until really about the mid-1600s when the compound microscope was invented by Robert Hooke. And then about 200 years later was the development of the cell theory. This wasn't just on Robert Hooke. He didn't live that long, obviously. But some big names within science. I'm not a huge name dropper, so I wouldn't spend a lot of time memorizing names. But I feel like Robert Hooke's an important one just because he did invent the microscope that you still are using today in lab. How many of you guys are using that digital histology DVD to help you with that first worksheet? Well, you don't. It's not required to purchase. You can use just the iPads if you go into Open Lab, and that might help you with some of those drawings. But we built that DVD with those images using technology that was developed in the mid-1600s, right? Because the optics have gotten better, the light has gotten better, but the principles are still the same. And so now we've just added new technology, but the real benefit of the histology on the DVD is more on the analytical side, evaluating tissues and looking at quantifying different features. We don't do that in this class, but that's where the field is today in research. So when you leave and you move on, more and more of the analysis of histology is happening off of a flat screen, not necessarily looking through binocular lenses. But we still use all the optics in order to generate the image. It's similar to what's happened with radiography. Right? How many of you have had an X-ray? That's what radiography is. The last time you had an X-ray, how many of you had the doctor put the film on a light box on the wall? A few. More of you probably just clicked on a screen and it was a big monitor and they cruised through the images on a monitor. That's what's happening with histology. But the principles are still the same. Just like X-ray is still the same. We've just digitized everything. So the cell theory has 4 basic tenets. Here are the 4 basic tenets that describe the properties of all cells. So number 1, they're the basic structural and functional unit of life, and they share generalized features. Like they have a nucleus. They have organelles intracellularly. They have a cytoskeleton architecture, which is what this picture is right here. Those are actually cells with their cytoskeleton, the inside framework of the cell. It's actually labeled with this fluorescent image. Organisms are all composed of 1 or more cells. Number 3, cell functions are carried out by these subcellular organelles stuffed inside. And then lastly, all cells arose from pre-existing cells. And this is sort of where we build everything from this point on. So let's take that last one, for example. An embryo. An embryo comes from cells, right? Gametes. One from the mother, one from the father. When you have cellular repair of your torn muscle or a broken bone, then those new cells, whether they're skeletal muscle cells or whether they're osteocytes or bone cells, they actually come from existing cells in the same tissue. So the cell theory can apply to a lot of aspects of medicine that you're going to come in contact with. So let's take a look back maybe a couple semesters of the contents inside a cell. Well, we know that there's a plasma or a cell membrane. It's the outer boundary of the cell. Remember how I said in the last lecture it's important to be able to compartmentalize and separate the inside from the outside. So the cell membrane or the plasma membrane, 2 words or 2 statements that really mean the same thing. They help to separate the inside from the outside. Who cares? Right? I already knew that. Well, it's become really important even later in the lecture when we start moving ions. and we start putting certain concentrations of ions on one side of the membrane and other concentrations of ions on the other side. Still, Keller, who cares? Well, in order to send a signal down a nerve, you actually leverage the gradients of those ions. What do you mean gradients? Well, concentration gradients. Like, oh gosh, I do remember something vaguely about concentration gradients and stuff moving down them or up them or with them or against. I can't remember, but I do remember them. Okay, so we're going to refresh your memory on what these concentration gradients are and why they're so important. But just flashing forward, if you want to send an electrical signal down a nerve, you use ions flowing down concentration gradients by opening certain channels in order for that nerve impulse to happen. Because those ions have an electrical charge. And when they move, they carry electricity, or they carry a differential in electricity. So being able to separate ions and thus separate charges is fundamentally important for carrying a nerve signal. Without a nerve signal, you don't have muscle contraction. Okay? Without a nerve signal, you don't breathe. Without a nerve signal, you don't move blood throughout the body. So they are important, and it all whittles down to the fundamental building blocks, which is at the cell level. That's why we spend time on it. I get a lot of students say, you know, I just want to take a class because I'm going to be a nurse or a doctor, and all this like cell crap and plant crap doesn't mean anything to me. And I'm telling you, it does. Many of our pharmaceuticals come from those plants. Plant crap, in quotes, right? So those pharmaceuticals you're prescribing, and the cells that receive them, it's like crap gets crap. You better know that crap. Okay? The outer boundary of the cell, it separates the interior from the exterior, and most oftentimes, they're not going to be the same. The extracellular fluid and the intracellular fluid is not going to be the same. We'll talk a little bit more about that. The cytoplasm is the cytosol, that's the fluid, and the organelles inside. And there's a great figure in your textbook, 3.5, that kind of highlights these things like what you see on the screen. Okay? So I want to make an analogy that the cell is kind of like a protein factory. What do I mean by that? Well, you manufacture products. Your cells do. Your cells manufacture products. Very soon after the first exam, we're going to start talking about the skin, or the integument. And you're going to see that the skin manufactures products that give structure to your skin that is the largest organ in your body. And it serves all sorts of important characteristics, like protecting you from the elements, preventing water from being lost. So, using the skin example, or bone, in lab this week, you're talking about skin and you're talking about the introduction to the skeletal system. Your bone cells, the osteocytes, they make the architecture that's the calcified bone. They also chew it up. So after the skin lecture, we'll come back in this class and we'll talk about the skeletal system and we'll talk about the physiology. I get questions from time to time, you know, Dr. Keller, why doesn't lab track exactly with lecture? Well, they're two completely separate classes, number one. And number two, you're going to spend maybe 4 or 5 weeks in muscles. You do not want me to lecture for 4 to 5 weeks on the physiology of muscles. Okay? It's not a 200-level course. So we build it appropriate for the level of this course. And so at times they come together and then they move apart and then they come together and they move apart. It's like this love affair. Okay? So we're close to coming together again, so you can look forward to that. All right, so this director, this manager, is the DNA. The DNA within the nucleus actually tells the cell what to do. Then this endomembrane transport system, those are the membranes within the cell. They're kind of like the assembly line. They move things down the assembly line as they're being manufactured. You've got support, administration, if you will. You have other organelles like the Golgi, okay, the ER. You have other cytosolic components that add supporting features to that product. Let's say that product is a protein. So the protein that's manufactured inside the cell is going to start by a code at the level of the nucleus and move throughout this assembly line. And the products are packaged. And interestingly enough, just like FedEx, UPS, or USPS, DHL, or you fill in the blank, there is actually a postal code attached to these proteins. There's actually a sequence on the protein that tells the cell where to send it. If you're making actin, that's a cytoskeletal filament. So the code says, keep it here. Right? This is a local delivery, if you will. And we locally deliver it to the inside of the cell. If it's collagen, you actually send it out. You send the collagen out, which is a structural protein of many, many, many structures, like skin and even bone. And that structural protein is transmitted to the tissue of interest. Okay, so the analogy of an assembly line and using a shipping code is actually very, very good. Collagen, like another one named as elastin, that's a stretchy protein. It starts as a liquid protein inside a vesicle, and then when it's exocytosed or exiting out of the cell, as soon as it comes in contact with components in the extracellular environment, when it dumps its contents, it actually cleaves that leader sequence and it spontaneously polymerizes. So it goes from a liquid to a solid. You're like, no way. It's like, yeah. Have you ever seen, it's kind of like, you're seeing like crazy glue that's in 2 tubes? You know, the double-chambered, double-barreled crazy glue? You following me? Some of you, you're not skilled enough with crazy glue, so you only buy the 1 tube. I understand. You know, you're like, right, I'm gluing my finger to my head. And so they do make this for the accelerated users. 2 tubes. Okay? There's 2 different liquids. One's an active catalyst and the other one's the base material. And when they come together at the nozzle, it creates an agent, a glue, that actually hardens pretty quickly. So it's kind of like that. So our cells are these manufacturing facilities, and they're actually super small. Right? I mean, think about this. We could be one giant cell, according to cell theory. Like, right? We could be one giant cell. But we're a multicellular organism, so why are the cells so small? And then the secondary question is, why so many? Right? So why are they small and why are there so many? If you look at the human cell, it's about 10 to 15 microns. And we have some other variations. Look at a frog egg. It's big, 1 to 3 millimeters. An ostrich egg is about 6 inches long. That's a single cell. And then the giant squid has a 39-foot long cell. So depending upon, you know, your definition of largest, is it length, right? Is it diameter? And we have a couple of different interesting versions in biology. Yeah.
Wait a second. How can a giant squid have a cell that's 39 feet long? You mean like their tentacles are all?
It's a neuron that's that long. So it goes like from the central nervous system all the way down.
Oh, so it's a neuron. It's not all the cells.
But a neuron's a cell. No, just a cell. A cell. Yeah, but a neuron is a cell. Good question. So why are they so small? Any questions? Any ideas? Any answers?
They were too big. Diffusion wouldn't be able to occur rapidly enough to sustain your cell.
If they were too big, it wouldn't be very efficient.
Yeah.
Totally agree. What else?
I was going to say surface area to volume ratio.
Surface area to volume ratio. Kind of another way, 3 ways of saying kind of the same thing. There's another reason. And it relates more to, because they're so small, we actually have a large number of them. What does the large number and small give us? Another advantage.
It helps for reproduction. Like, if one cell dies, it regenerates, or there's multiple other cells that can help carry on that function.
Absolutely. If I was a single-celled organism and I just had cellular death right now, but just a moment, right? And then it would take a while before me to come back online. It'd be super disruptive to lecture. So fortunately, cellular regeneration is happening right in front of you as we speak. And I don't get to miss a beat. Right? I make new skin cells. I slough off the old ones. I leave a little behind. Okay? Just kind of sprinkle them over there. And I just keep lecturing. But don't be so grossed out because you're doing the same thing. Okay? How many cells do you think we could extract out of that seat cushion you're sitting on? If you want to bring a plastic bag this Wednesday, okay, so I agree. So reproduction, efficiency, what else? There's a third one.
Complexity, like intellectual complexity, body.
So complexity, I do like that. Another way to spin that might be specialization. Okay? If I was just one giant cell, how could I move blood? How could I respire? How could I exchange gases? How could I filter? How could I have neuronal complexity? So now all of a sudden you have specialization. And we're going to look throughout this semester, and you're going to look throughout next semester in 202 about the different types of complexity based upon the different types of cells. Well, they're all cells. Like the neuron from the squid, we have neurons too. They're not quite that long, but they perform a very specialized function versus a contractile muscle cell. A fibroblast in the skin, still a cell. A little bigger, maybe 25 microns in diameter. Red blood cell, a little smaller, right? Maybe about 7 to 9 microns in diameter. So the red blood cell, smaller, carries oxygen. The fibroblast is much bigger, and it produces vast amounts of collagen. So we have specialization as a result of these cells. They're small, so we need microscopes to see them. That's why you look through a microscope, or you're sifting through images that have been generated on that DVD from a microscope. Because you need magnification in order to see them. Well, sometimes passing light isn't the best way to see them. We actually transmit electrons. Okay? It sounds super space age, but it happens right here on this campus. Electron microscopy actually, instead of shining visible light, you actually coat the sample with an electron-dense material, and you shoot electrons at it, and it bounces off the surface. You collect them on a detector, and that recreates an image. So light microscopy can get down to the stuff that you're seeing. Electron microscopy gets us down even farther. Much, much higher magnification. Okay, so hopefully I've convinced you why cells are so important. So let's talk about their plasma membrane, and let's talk about some of the features. This is all review. I'm just trying to package it in a context that might make a little sense to what we're talking about now with the human body. Different functions of the plasma membrane, or the PM, are shown on this slide. Barrier function. Prevent water loss. Prevent microbes from coming in. We'll see in the skin lecture, after the exam, when we talk about wound healing, or wound care. The biggest challenge that we have for patients that are maybe 70, 80, 90% burned, and they've lost their skin coverage, number one is we worry about water loss and dehydration. Number two, what's the second thing we worry about? Infection. So you worry about losing things, number one, then you worry about things coming in. So that barrier function of the skin is really important. So this week when you look at the different layers of the epidermis, keep in mind, why do I care so much about this? Well, when it is compromised, you have some big possible issues. And you're going to be dealing with patients like type 2 diabetic patients that have chronic non-healing wounds where they don't have complete skin coverage like over the bottom of their heel or their big toe. And if you're not able to restore that wound, that coverage, that barrier, they might lose that limb. Maybe not the limb, they might lose the toe or the foot. Hopefully it's just below the knee, because then our prosthetics are a lot better when we don't have to cross the knee. They serve a gatekeeper function. They allow certain things in, and they keep certain things out. They support the shape or the architecture of the cell. You've been studying, we're going to talk about it today in tissues, you've been studying the different shapes of the cells, whether they're flattened like squamous, or they're cuboidal, or the columnar. Want to bet that the shape or the architecture helps to determine how they work or their function? Absolutely. You're going to see many times in lecture, in this material, as well as in laboratory, that the architectural components give rise to the functional physiology, or structure gives rise to function, as we like to say. They also help cells communicate from one to another. In 202 next semester with cardiac, do you think it's important that the cell in the heart next to the other cell, no, me first, me first. Which one contracts first? Well, there has to be a very systematic contraction, and they have to be able to communicate. Those are those intercalated discs that you've been looking at under the microscope. They help with cellular communication from one cell to another, so they know which one contracts first. If they get out of sequence, then we have some arrhythmias. Okay? And those patients of yours would either have a pacemaker, likely a pacemaker with a defibrillator, okay? Or we have things like heart block. All right, you can see the membrane here. This is that phospholipid bilayer. And you can see these transmembrane proteins that span across the plasma membrane. So if we look more specifically at the plasma membrane, we see this cartoon drawing here. We see some architectural helical structures showing different orders of the protein that we covered in the last lecture. And we see that we have some saccharide molecules, some sugar molecules that we covered in the last lecture that are stuck on the outside. Many proteins that have an exposure to the outside environment have a sugar residue on them. And there's this sort of layer on the outside of the cell. Most cells have this layer, this coating around them. We call it a glycocalyx. And this glycocalyx is kind of like a sugar-coated cell. It's really good with milk, maybe a little bit of fruit, you know. So these sugar-coated cells is very common in the body because it's in an aqueous environment, and sugar is hydrophilic. It's readily dissolved in water. So the phospholipid bilayer has these proteins embedded in it. And the hydrophobic region is in the middle, and the hydrophilic region is either sticking out or sticking in. We've described this in previous classes as this fluid mosaic model, or this fluid mosaic theory. What that means is I like to envision it like a swimming pool. You have the surface of a swimming pool, and the top portion of the phospholipid bilayer was a styrofoam ball. And you covered the entire surface of the swimming pool with styrofoam balls. You could take any one ball and kind of move it from the deep end to the shallow end without ever losing contact with the surface of the water. Okay, so it is fluid like that. And then there may be, a few of them that you paint blue and a few of them you paint green. And you can imagine that those might represent structures that are embedded in the membrane. Okay? Of course, they're tethered underneath the water to the bottom of the pool. So if you tug on one, it's going to tug on the bottom of the pool as well, because there's an internal architecture. But you can appreciate how the surface of the pool with these spheres has a level of fluid-like behavior. So our cells aren't rigid, right? They can adapt. This is a transmission electron micrograph. And this is 100 nanometers. This is the phospholipid bilayer. If you look carefully, you can kind of see a darker line on the bottom, a lighter region in the middle, and then a darker line on top. And the two darker lines are the polar heads. And the lighter region in the middle is the hydrophobic tails. So this phospholipid bilayer limits what things can come in and out. This is one of the important attributes of it in our cells. If we're talking about neuronal cells, then we're going to limit when sodium enters. Because when sodium enters a neuron, you stimulate an action potential if it reaches threshold. We'll talk more about that later in the semester. But certain things can cross and certain things cannot cross, so we say that it's selectively permeable. So if we want to get other things that cannot cross into the cell, how do we do that? Any ideas?
Protein?
Transport protein, a carrier molecule. Okay, so let's talk specifically about these proteins. These proteins are called either integral proteins or transmembrane proteins. They're integral, meaning they're into or incorporated into the membrane. In some cases, they span all the way across, so they're called transmembrane. That's what that word, that prefix trans means across. Okay? So here's an example of a transmembrane protein that has a hole down the middle It's open on the exterior aspect, which is the extracellular fluid, and it's right now closed on the inside, or the intracellular fluid, so it is not allowing stuff to move through. However, we can actually gate this and control its opening and closing, and we can do that using electricity, and if you call a voltage-gated channel. We can control it using a ligand, like a neurotransmitter, and we would call it a ligand-gated channel. Or we could gate it mechanically, like if you put pressure on it, it's going to respond. Like your Pacinian corpuscle, right? Or your Meissner's corpuscle. They're mechanoreceptors, meaning that the gated channel is sensitive to pressure, like a pressure transducer. You put pressure on it, it opens the gate. The gate opens, sodium comes in, stimulates a signal. Now you don't know the magic of what the signal is, but we'll get there later in the semester. The peripheral proteins are associated with the membrane, but they may not be embedded into it. So we call them peripheral. So we have integral that are embedded in. We have transmembrane that go all the way across, and then we have peripherals. We kind of have three types of proteins associated with the plasma membrane. I already told you that most of these are glycoproteins, meaning they have a sugar residue on the outer side. There's that word. I'm surprised nobody asked me how to spell it. You guys must be above average. Or you just didn't want to admit that you didn't know how to spell it, because everybody knows how to spell that. Of course. Right? Yeah, I mean, come on, how complicated is that word? So Glycocalyx, this is the sugar coating on the extracellular side of the plasma membrane. The sugar likes to exist there because it's hydrophilic, and that's an aqueous environment. Look at all the different functions of these proteins. We've got transportation, recognition, enzymatic activity, cell-to-cell linkage or connections, receptors, or cytoskeleton attachment. So these proteins are critically important for the normal function of that particular cell. The cell would not be alive if it wasn't for them. Well, we also have sort of other ways to classify proteins. We can classify them as structural in of themselves or functional. So if we look at our first structural proteins, and I kind of alluded to these already, these are considered fibrous. They are not very soluble in water. Does that make sense?
Yeah.
Why?
Once they dissolve in the cytoplasm, you'd be in big trouble.
Yeah, you would just, right, just kind of fall apart. So they're not very soluble. That makes sense. So here's a perfect example of how the architectural structure gives rise to its function. If you're building a structural protein in an aqueous environment, it's like putting a bridge across the bay. And it's like, I think we're going to use water-degrading concrete. It's earth-friendly. It's like, brilliant, you're fired. Next. So this makes total sense. They're long, they're rigid, they're rod-like. They give structure not just outside or the cell or inside the cell, but also outside the cell. So because both the outside and the inside are aqueous environments. The cytoskeleton example would be actin. The extracellular example would be collagen, the one that I mentioned earlier. Here's a transmission electron micrograph. You wouldn't be able to see this level of detail under a light microscope like what you have in the lab. You got to shoot electrons at it. Okay, go. This reminds me, have anybody ever seen like a overpass or a bridge being built with a lot of concrete? You ever drive by, or even here on campus, with, there's like 17 buildings going up all of a sudden. And you drive by and you see like these like steel rods coming out and bending, and that's rebar, right? And that is, it's like a ribbed rod, and they can shape it in different formats. Then they pour the concrete over top of that, and that's the structural innards that give the strength to the building or the bridge, okay, or the overpass. Without that, concrete's extremely brittle. Okay? So that's what this reminds me of, is they look like rebar. That's what our tissues are made up of, are these structural rod-like proteins. They're considered fibrous, not very soluble in water. You can solubilize them in other things, Most of them are soluble in acids for experimental purposes, not like, nobody has, acid fluid within them, which would be kind of cool, but be like an X-Man. Okay. Any X-Men in the audience? Okay, just check. I don't want to offend you if you are here. So, okay. The second category would be considered functional. So structural, not that they don't have a function, but structure, their function really is their structure. The functional means they're going to have a real physiologic role. Now, their structure is going to give rise to that functional role, just like the structural proteins. That's like using the definition to define the word, right? And vice versa. These are considered globular. And so the example that we used in a previous lecture was hemoglobin. It's a globular protein. Many, many times they have a soluble aspect. Right? So hemoglobin is soluble to some level within the blood. It's an aqueous environment. You want it to go into solution. So it floats around. It doesn't get stuck. But usually they have a limit of their solubility because the polar groups are facing out. That allows it to be soluble. And it hides the nonpolar groups, which are on the inside. So it's a little bit of a trick. Okay? It's a trick on how you get something insoluble into solution. Is you put the polar aspects on the outside. This is how detergents work. That's how detergents make grease go into solution. Same principle here. Okay? So you can kind of see the units, the beta chain here, and the alpha chain here, and there's two of each. And then within the alpha and the beta chains are these heme groups that have an iron. That iron is what has that affinity for oxygen, and that binds, and I told you last time, 4 maximum oxygen molecules will bind to a hemoglobin molecule. We have a carrying capacity of 4 oxygen molecules per hemoglobin. They're compact, and they have these active sites. That heme group is considered an active site. So globular or functional proteins serve a big physiologic role that gives us an advantage about something. So let's talk back a little bit more about this protein factory analogy. And I want to see if you can tell me the order of starting with the nucleus. If we were sending a protein product out of the cell, where would it go next? Then where would it go after that? And where would it go fourth, fifth, so on and so forth? You're starting at the nucleus, you're going outside the cell. Okay? There's cameras on you right now, evaluating who's participating. Anybody old enough to remember the Gong Show? Sweet. Oh, one dude in the back? Right on. From one old guy to another. Nice. Why is that relevant? Someone's like, what? I don't even know what that is. It's kind of, like, if you messed up, there's this giant gong, right?
That mean individual functions?
Yeah, you're building a protein. You start with the nucleus. That's the starting point. You're going to end outside the cell. It goes from the nucleus to what next? And then to what? And then to what? And eventually it makes it outside the cell.
Are the functions of the organelle going to be on the exam?
Yes. You guys set? Let's review, right? You got it down. This is easy. Okay, I see some, these kind of looks. It's like, is that painful? Wow. Someone just pulled a toenail off, or maybe we should review this week. Yeah.
Are you looking for answers, or are you writing it down?
You're writing it down. Yeah, you're writing it down. Like, what do you want me to do with this info?
Yeah.
We're going to go over it. You guys got it written down? All right, we're starting with the nucleus. What's the next structure? Where does it go? Ribosome. From the ribosome, where does it go? To the rough ER. From the rough ER, where does it go? You need a vesicle to get it there. So a vesicle, then to the Golgi. Okay, then a secondary vesicle system is the lysosome that sends it to the plasma membrane. One more time? Yeah. Well, you had it on your list. I thought you just double. You're just like, yep. Oh, I just missed one. Right? How many of you that was the case? Okay, so 7 of you. Okay, cool. So 393 of you need to look at this. Will these be on the exam? Totally. Do you need to know what they do? Yes. This is review. It was a prerequisite, so technically you already know this. Sometimes I get questions like, why are we going over this? Because 7 of you know it. That's why. Nucleus, ribosome, rough ER, vesicle, Golgi, lysosome, plasma membrane. If you disagree about the lysosome, because it has specific function, you wanted to use vesicle in both places, fine, I would agree with that. Question?
Only these organelles or all of them?
Only the one, what I cover in lecture is fair game. So these organelles. Okay? Yeah, that's my attempt to be as fair as possible. If I have time to cover it, you should know it well. If I don't get to it, then I won't put a question on there. Other than maybe what's the NOAA weather patterns for the San Juans and the Wasatch on Friday. Just kidding. But that's my exam, not yours. All right. More review. You ready for more review? Because review is so much fun. Let's talk about where we get our energy. Cellular respiration. See if you can do this. If you printed this out, see if you can identify. There's 3 labels. There's a label here, there's a label here, and a label here. And the labels are glycolysis, Krebs, and the ETC. I'm looking for where do these things take place. So if you're confused by the picture, like when I have this on the screen in my office at home, and the girls walk by, they're like, "Are you studying that big finger again?" Because they know I teach A&P. Like, "Oh, that finger looks bad. It looks really messed up right there." You know? Like, "No, honey, that's the mitochondria." "What's the mitochondria? Is that on the finger?" Like, well, it's hard to answer that question. I don't know how to answer it. Do you want any other jokes, sweetheart? I'm running low on jokes. All right. So if you're distracted by the giant finger, then just answer this on your paper. Where does glycolysis take place? Where does the Krebs cycle take place? Where does the electron transport chain take place? Don't shout it out. Write it down. Okay? If you want to converse with your neighbor, that's fine, but not next Monday. Do your own work.
Yeah.
All review, super easy. Make Dr. Pollack happy. You know this. Dr. Holloway would be proud. All set? Should I reveal? Where does glycolysis take place? Cytoplasm of the cell. Where does the Krebs cycle take place? Mitochondrial matrix. And the ETC? The inner space of the mitochondria, right? So if you reveal these, you can kind of appreciate glycolysis, right, happening within the cytoplasm. Because the mitochondria is this structure. This is the cytoplasm. And then here you've got your Krebs cycle, and then you have your electron transport chain. So, continuing on this theme, I want to kind of remind you that we have these substrates, and we've covered these substrates. These are all 3 organic types of molecules, right? Proteins, carbohydrates, and fats or lipids. Now, all of them can generate ATP. And depending upon your activity and your substrate demand or availability, you may switch from one form of energy to another. Those of you that are going exercise science, you'll study this a lot. But even if you're not going exercise science, it's important for you to understand how this works. Right? I mean, even little things like fad diets. This would be helpful from a nutritional standpoint to be able to explain to a patient, right, in your office, what that Atkins diet is actually doing. Okay, so that's how it relates. This person published tons of books on this diet. And it's like, I've had this slide for years, man. I mean, I don't know, this is nothing new, but I didn't write as well as him, apparently. Okay? So you look at the different building blocks, and they shuttle into either a glycolysis pathway, and pyruvate in the presence of oxygen feed into citric acid cycle, generate additional ATP. The most ATP comes out of the electron transport chain. There was a question.
For the glycolysis, would you accept that it occurs in the cytosol as well as cytoplasm?
Yeah, that's fine. Yep. Glycolysis, what you're asking about.
Yeah.
Yes. So, along those lines, let's define these. So, sometimes we do these bonus opportunities in class, and this could be one. I toyed with the idea. Where I want you to define it. I'm not going to collect this. You don't have to tear it out. I heard a couple tears. You're like, oh man, I just killed a tree. Plant it. It'll grow back. Glycolysis. Tell me what it means. Glycogenolysis, gluconeogenesis, and glycogen. Define each one of the four. Glycolysis should be easy. We just went over it. $200 on the screen right there, right? 4 $50 words. Yeah, they're worth $50 at the minimum.
You're going to give us $50?
No, I'm just saying that's the value you're getting today.
Okay.
All right, who's got a definition for glycolysis? Right here.
Oxidation of glucose into 2 3-carbon pyruvates.
Ooh, very nice. Nicely done. Okay. Anybody else have a different definition? I mean, one not as basic. I mean, that was so, Anybody off the sidewalk could have said that. Anybody else have a glycolysis definition? Yes? The breakdown of glucose to do what? Why would you just break it down for what purpose? To make ATP. ATP is the fancy what? What's the word you used? 3-carbon molecule? 2 3-carbon molecules?
Pyruvates.
Right. Or pyruvate, excuse me. So ATP. So glycolysis manufactures ATP. Breakdown of glucose to make ATP. Glycogenolysis. Who's got one of those? Somebody else other than, so our back row is really, our back front row, that's kind of weird, but right there. Okay, breaking down glycogen, or the hydrolysis of glycogen to make glucose. So that you can put it into glycolysis and then yield ATP and pyruvate. The pyruvate would move on in the presence of oxygen, right? Converting it to acetyl-CoA and coming into the Krebs cycle. Okay, our third one. Gluconeogenesis. This one might be more of a $75 word. It's kind of longer. It's worth that. Not that I'm handing that dollar amount out. Gluconeogenesis. Anybody else want to play this game other than the 3 participants? In the very back.
It's the conversion of protein to glucose, right?
It is. It's the conversion of protein to glucose. It's really commonly referred to as the production of new glucose from scratch. Okay, but you would use building blocks like at the level of the liver. You know, take proteins to convert and create glucose without breaking down glycogen. You could also do it from glycerol or lactate. So amino acids are proteins, glycerol or lactate. as the starting material in the liver, and you make new glucose. So if you break the word down, glucose, new, beginning. Gluconeogenesis. Glucose, new, beginning. Glycogen. What is glycogen? A couple of different definitions I'm looking for here. Yep, it's the compound molecule of glucose, or another way of saying that would be what? The storage form of glucose, or a monosaccharide, disaccharide, or polysaccharide? Polysaccharide. So it is a polymer of sugars? Yes. So there's like 4 or 5 definitions for you, all that point to glycogen. You with me? All right. How did you do? Compare with your neighbor. Yeah. The question is gluconeogenesis. The starting material for gluconeogenesis could be amino acids or protein, as the gentleman in the back said. It could come from a fat source, or it could come from it's just a non-carbohydrate source. So fat or protein making new glucose. A non-carbohydrate source. All right, try this possible test question. You perform a chemical analysis of a bone cell. You'll learn this week, we call that an osteocyte. You'll learn that in lab. You find out it contains large amounts of pyruvate. You determine that what has taken place, or what has occurred. Gluconeogenesis, the Krebs cycle, glycogenolysis, 2 of the above, or none of the above, or none of these. What's the answer?
I think none because it sounds like anaerobic respiration.
It is exactly that. So the answer would be none of the above because what? Pyruvate is a byproduct of anaerobic respiration or glycolysis. Okay? Perfect. Question?
So is anaerobic respiration another word for glycolysis?
Yes. Yeah. Glycolysis is an anaerobic production of ATP. Okay? Yeah.
Wait, what is pyruvate glycolysis?
Glycolysis. Pyruvate is a byproduct of glycolysis. Okay? All right, let's look at moving things across a membrane. So some of those terms might be part review and maybe mostly new. So we're starting to get into some compounded new stuff. But that's what we're after, is to get this basic framework of familiarity so we're all on the same page with the review material. From here we can spring forward and start getting into the new stuff. But the first exam tends to be more gentle, meaning it's not as bad because a lot of you are familiar with the material. Okay, then we get a higher level of difficulty on exam 2. Exam 3 and 4 are the most difficult for the semester. That's the most difficult material. And then exam 5, the last one, we kind of come down on the difficulty level again. So that's kind of the course. Right? Yes?
I'm wondering, for the exams, are you going to have material from both this lecture and from the laboratory, or just from the lecture?
Just from lecture for the exam. I'll talk a little bit more about the lecture format, the exam format in lecture on Wednesday. But generally 50 questions, multiple choice, or multiple guess, depending upon how it works for you that day. It's all material from lecture. I just reference lab a lot in lecture so that you feel like you haven't lost touch with what's happening with this other component of similar material. So we look at membrane transport. We already said that we've got the ability to separate the inside from the outside of the cell. We already said in previous lectures that the solvent plus the solute equals a solution. So in our case, we're in a saline solution, and so it's water with sodium chloride. And water with sodium chloride makes a saline type of solution, which is extremely friendly for life. In cells, that second bullet point says the extracellular concentration does not equal the intracellular concentration. That's how you would read that. In fact, when it is equal, usually that cell is dead. The cell spends a lot of energy creating differences of gradients in order to facilitate different processes. One thing that it does, and why it does that, is so that you create a concentration gradient that you've learned about in previous classes. That concentration gradient allows for a high region of a sodium on the outside of the cell to pour into the cell quickly when you open up a transporter that allows for it to move. Then there's another transport mechanism that requires ATP, known as the sodium-potassium ATPase that you've heard about. And that works and spends energy, valuable energy, pushing 3 sodiums out of the cell and bringing 2 potassiums in. That makes the outside sodium concentration high and the inside potassium concentration high. We'll go over that again in case you are writing feverishly. But this diffusion definition is the movement down the gradient. Down means from high to low. Up would be the opposite direction, from low to high, or against. So sometimes students get these terminologies mixed up. Down the concentration means you go from an area of high concentration to an area of low concentration. The way water flows. Okay? So if we talk about transporting stuff across the membrane, we have 2 main categories. We call them passive transport and active transport. Passive transport, no energy required. It goes from high to low. It moves down its concentration gradient. No energy required. Active transport takes energy, usually in the format of ATP, and it's going to go in the opposite direction of passive. It's going to go from low to high. Or it's going to move up its concentration gradient. Or it's going to go against its concentration gradient. Make sense? Okay. Interestingly, sugars, ions, amino acids can move via passive or active depending upon what the concentration gradient is. What that statement means is if there isn't a concentration gradient set up, you can actually use a transporter and burn energy. So you're not like stuck going, oh crap, I got to wait around for a gradient to show up. You could actually still move it, you're just going to burn energy to do it. You with me? This second little note says, in certain cases, these things are really big that you want to move. And so that would be considered bulk transport. That's like, for example, if you want to move proteins. Like in the example we were talking about a little bit earlier. And when you do bulk transport, a lot of times you house them in membrane vesicles. This figure down here is showing outside, you probably can't read this, this area up on the top part is the extracellular fluid, the ECF, and the area in the bottom is the ICF, or the intracellular fluid. And you can see, let's pretend this is a sodium molecule. Here is a transmembrane protein that sodium molecule binds to. The protein goes through conformational change, opens the channel, and then the sodium is dumped in. And if no ATP is burned, then that would be a passive transport. Okay? Different types of passive transport. We have what we call simple diffusion. Simple diffusion is relevant for small molecules or substances, usually ones that are lipid soluble because they have to go through a membrane. Examples include oxygen, carbon dioxide, and fat-soluble vitamins. Now, osmosis is a special case of diffusion. Osmosis is a special case of diffusion because it talks about water movement. But no energy is required. So it's still diffusion. It's just a special case because you're talking about water moving instead of talking about a substance or a small molecule moving. So let's explain. 2 pictures. You have a beaker with a dotted line down the middle, and this is representing one cell, cell 1, and cell 2. And that dotted line is a semi-permeable membrane, like the membrane between 2 adjacent cells. And the small little spheres are water molecules. And the large orange spheres are large protein like albumin, huge protein. And the membrane is not big enough, the diameter of the hole, to fit the albumin. So if it was in high concentration here, it would want to move this way, but it can't. So instead, water moves. And water follows solutes. Remember that statement? So water moves to where there's a high concentration of solutes. So 30 minutes later, you see the level of water in this side is lower because the water moved to the area here via osmosis. So water movement is a special case or a special type of diffusion. How is this relevant to this class? Like, yeah, I remember that bit. Well, let's look at a very important cell in a solution. Okay? Anybody know what these pictures are of? Red blood cell. Okay? The one in the middle. This is what it's supposed to look like. That's in an isotonic solution. Iso meaning same. Isotonic solution. You talk about the tonicity of the solution. So in an isotonic solution, this would be a 0.9% saline solution or sodium chloride solution. That's typically what the bag is on the IV rack that you deliver to a patient. This is why you don't put pure water into the veins. Okay? You could cause some big problems. Okay? So I will quiz you if I'm old and on a bed and you're hanging a bag. I'm going to want to read the label. Okay? Or I'm going to ask you what grade you got in my class. Either one will be fine. Right? They're both a number. Okay. 0.9% sodium chloride. Biconcave, it has a little dimple on both sides. This increases the surface area and therefore allows oxygen carrying capacity of the red blood cell to be superior. Now, look at this one. What solution do you think on the left is? Hypotonic. Right? What's hypotonic mean? It's pretty much pure water, right? So if you put the cell in a hypotonic pure water, which direction does water move? Into the cell, and now the cell swells. Over here, this is what? The opposite. Hypertonic solution, right? So instead of pure water, that would be like 2% sodium chloride. And so what happens is the water leaves to go where there's a higher concentration of salt. And then the red blood cell has what we call a crenated appearance. So water moves down its concentration gradient until it reaches equilibrium. Many times in this situation, you may lyse the red blood cell. So if you hang a bag of water instead of 0.9% saline, you could actually lyse the patient's red blood cells and kill them. That would not be a good day for anybody. Okay? Yeah, you probably would get sued. Okay? All right, let's look at this. This red blood cell has been put into what type of solution? Isotonic A, raise your hand if you want that one. Hypertonic B, raise your hand if you want that one. Hypotonic C, or you cannot determine from the information given. Okay. The B's have it, right? Hypertonic solution, which is this picture right here. All right, let's talk about facilitated passive diffusion. This is protein assisted, meaning that you need a carrier to get it across. A lot of times, this is what's involved when you want to move amino acids or you want to move sugar, glucose. A lot of times there are channels or pores that exist. We can regulate those. We can regulate them with electricity, called voltage regulated, or we can regulate them with a ligand called ligand regulated. Example that we'll see in subsequent weeks is the nerve cell. And this was that picture that I showed you in much smaller format on a previous slide. So What do you think happens in this situation of question 1, question 2, or question 3? What happens in 1? You have a 10% glucose concentration inside the cell, and you drop it into a 20% glucose solution. What happens? The cell shrinks. What happens if you put a 10% glucose into a 10% glucose? Nothing. No net change. It's another way of asking this red blood cell question. What happens if you put 10% glucose into distilled water? The cell swells, right? Well, when we continue on with this idea of transporting, we've got passive, and now we're going to talk about our active in the remaining time that we have. Active transport moves stuff against its concentration gradient, or moves solute up its concentration gradient, or from low to high. All of those would be accurate statements. It requires energy because you have to input energy in the system to go against the concentration gradient. A lot of times we use protein called pumps. And the one that you're most familiar with, I alluded to earlier, here's a picture of it. This is a phospholipid bilayer. Here's our sodium-potassium ATPase pump. And it's pumping out the orange spheres. 3 sodiums go out, and 2 blue spheres, 2 potassiums go in for every cycle of the pump. And it burns ATP in the process of doing that. So this naturally creates high sodium outside the cell and low sodium inside the cell. And high potassium inside the cell and low potassium outside the cell. If we look at secondary active transport, this one has an indirect consumption of ATP. So you can see down here at the basal surface of the cell, we have our sodium-potassium ATPase working, right? Creating a gradient of sodium. Now if you look up here at the apical surface, sodium is high, and here's a sodium glucose transporter, an SGLT. So it's going to co-transport sodium with glucose. So as it opens its doors and sodium wants to come in, because of the gradient set up down here burning ATP, glucose is going to hitch a ride. So this is called a secondary active transporter. Right? It's still active because ATP is consumed, although it's consumed indirectly in order to set up the gradient that glucose is going to use in order to get into the cell. So what does the sodium-potassium pump do? It maintains our resting membrane potential. It creates higher potassium inside the cell, and it creates higher sodium outside the cell. This actually moving 3 positive charges out and only 2 positive charges in, that creates a net of -1, right? 3 minus 2. I know it's biology, but I mean, you could use your phone for that if you need. 3 minus 2 is a net -1. So that means there is a differential of charge across the membrane as a result of this. And we're going to use that later in the semester. It uses a lot of ATP to do this. 30 to 50% of all the ATP that you consume is to run this pump. And that creates heat. That actually establishes your resting temperature, your 98.6 degrees Fahrenheit or 37 degrees Celsius body temperature. Okay, our last slide, and then we'll break for the day. Bulk transport. If a macromolecule is too big, you actually have to use a facilitated endocytosis or exocytosis. Endo brings it in, exocytosis puts it out. If it's large, we usually call this phagocytosis. That means cell eating. If it's a smaller molecule, we call it penocytosis. That means cell drinking. And you can see this is an example of a clathrin-coated pit bringing in lipid. And so it uses this molecule It sort of invaginates the membrane. These are transmission electron micrographs up here. This is actually the cartoon that matches it below. It pinches off and brings it into the cell, either in a phagocytotic process if it's large, or a pinocytotic process if it's small. Okay, that's low-density lipoprotein coming into the cell. I'll see you guys on Wednesday. Don't forget to do quiz and worksheet number 1.